and I'm really excited to have Benjamin Garcia here. So Professor Ben Garcia is a chair of the Department of Biophysics at WashU, and he's a Raymond Whitkoff Distinguished Professor. And before at WashU, and before that, he was the presidential professor at UPenn, and before that, he was a faculty in Princeton. Ben has won numerous awards, which will take me 30 minutes to go through. But some of the coolest things he's won are he won the NIH New Innovator Award, the PCASI Award, the Presidential Early Career Award for Scientists and Engineers. He was a Sloan Fellow, and he's also a Fellow of the Royal Society of Chemistry. And Ben's lab has done some really cool work using mass spec to study epigenome, protein modifications on histones. And I'm really looking forward to your seminar.
Okay, thank you very much for that really kind introduction and the invite to come share some of our work over the last year or two that we've been able to accomplish. And what I'm hoping to do today is give everybody, in the first half of the talk, kind of a crash course or update on some of the technologies that we've been developing over the last few years. And then kind of second part of the talk will focus on how we're applying these technologies to understanding epigenetic mechanisms in a very specific sarcoma cancer. And so just to get everybody on the same page with regards to background, I know not everybody studies chromatin or epigenetics. That's okay, nobody's perfect, just kidding. But we have large amounts of DNA, and the way that nature kind of is able to take this DNA and put it in a very small area in the nucleus is by wrapping the DNA around a ball of proteins called histones. And about 146 base pair of DNA will complex with 2 copies of the 4 core histones and make a nucleosome that's the basic repeating unit chromatin. And the nucleosomes will be connected together. Some will be in more densely aggregated regions versus more open regions of the genome, but ultimately this will make our chromatin fiber and our chromosome. And these histone proteins are very important for this process. And at first it was thought that they were more structural proteins involved in just, the structural aspects of chromosomes. But then, researchers over several decades have discovered that these histones are highly post-translationally modified. And in fact, the modification kind of landscape of these histones is quite immense. And I would probably go on a limb and say that they're probably the most highly modified protein in the human body. And so here's the 4 core histones, H3, H4, H2A, and H2B, and the linker histone H1 that's in between nucleosomes. And you can see here, there's many different modification sites spread across the histone. And this is probably out of date by now because this is several years ago. But it just gives you an idea of all the different modification sites. There's some sites that have multiple types of modifications that can be found. And as you can see here, the chemical diversity of these modifications is quite high. Definitely things that you've heard before, phosphorylation or methylation of lysines, things like that. But there's a lot of really interesting modifications that have been found and are continuing to be found on histones, such as ADP ribosylation, sugars, and a whole series of fatty acyl modifications. And these modifications that many of them, not all of them, but many of them are on the N-terminal tails that stick out of the nucleosomes. These N-terminal modifications have been linked to gene expression patterns. And so again, you're kind of seeing the core histone tails in cartoon form with some of the more abundant modifications that you'll see. And in general, histone acetylation has been found to be linked to gene activation. Histone methylation, it's a little bit more complex. It comes as monodi trimethylation, so different degrees of methylation, and it could be linked to gene activation or silencing. For example, lysine 4 methylation is found on the promoter regions of active genes on histone H3, but if you go a few amino acids down, the same type of modification, lysine methylation, is actually on K9 of the same proteins, actually found on the parts of the genome that are silenced. And so it's not only the modifications and what type, but where they're at on the sequence that really matters. And because of kind of these relationships, many theories and kind of hypotheses have been put together to try to understand how histone modifications affect gene expression patterns. And one of these is like the histone code or histone PTM code type of hypothesis that basically says that these modifications act as a binding platform which recruit other proteins, and this leads to gene activation or silencing. And in support, several proteins that bind specific histone modifications have been found, and these are kind of like some of the first steps that will then turn on or turn off a gene. And histone modifications, along with non-coding RNAs and DNA methylation, make up these epigenetic mechanisms. And epigenetics kind of refers to gene expression patterns and cellular phenotypes that can be passed down from one generation to the next, and that not require any type of underlying change to the DNA sequence. So there's no DNA mutations. And as you can imagine, there's a lot of areas of biology that this could become really important, especially developmental biology. So if you have a stem cell and it's going to convert to many different cell types and tissues, organs, and a whole organism, epigenetics has been shown to play a big role here in control the gene expression patterns to differentiate cells to different lineages. But there's a lot of real interest in understanding epigenetics in human disease as well. And that's because with the huge amount of genomic sequencing data that's been coming out over the last several years, many mutations to histone-modifying enzymes, proteins that interact with histones, or even the histones themselves have been discovered. And so there's many companies, large companies that all have epigenetic oncology divisions where they're creating kind of small molecule inhibitors as kind of next generation therapies for various cancers and other disorders. And there's just a lot of interest in understanding how epigenetics may play a role in different biological processes as well. And you know that your field is a really important one when it makes it into the really blockbuster movies. And so what you're seeing is a picture still from one of the Spider-Man movies from a long time ago. So there's the board is full of all the science and math that required to create a superhuman spider. And if you look at the board, you'll see a lot of things that are familiar, but you'll see something right here that looks very, very familiar, at least to me it does. It's a nucleosome with histone H3 K27 trimethylation on it. So whoever's in charge of this board knew that epigenetics was just a really important field that probably plays a big role in obtaining superhuman powers. And there's a lot of different ways to study histone modifications. Traditional approaches with site-specific antibodies. And so these are antibodies that recognize more or less one site at a time. However, there's issues, right? So when you're doing Western blots, you often see multiple bands that it can recognize, and that's cross-reactivity. So the antibody may not be super specific, and it's a big problem with histones because a lot of the histone sequences are very similar and highly modified. So you often get cross-reactivity that occurs. But a bigger issue is what you really don't see. And so here's a peptide ELISA assay. So you have these synthetic peptides, they all have serine 10 phosphorylation. Here's a monoclonal antibody that recognizes that serine 10 phosphorylation. The last two peptides have a modification right adjacent to that phosphorylation. The antibody is really great at recognizing phosphorylation by itself, but has trouble recognizing the duly modified peptide. And this occurs in vivo. And so that's why we kind of favor using mass spectrometry analysis, because we think it's a little bit less biased approach and we can do things much more quantitatively than with immunoassay or antibody-based type of methods. And over the years, my lab has been developing a lot of approaches for just understanding a lot of different aspects of histone PTM biology, trying to make these analyses a lot more quantitative and high throughput, trying to understand which combinations of modifications occur together, how fast these modifications get put on and off. Those are things that we've been, coupling mass spec with in-cell labeling, as well as coupling, genomic approaches with mass specs to, mass spec readout to try to get to, looking at your favorite gene and the proteins and the histone marks that are present in both healthy and disease states. So I'll talk a little bit about some of this technology we've been developing. First, kind of our approach is to look at the combinatorial histone PTMs that are present. And so, from our data and others, one single histone modification site almost never occurs by itself. It's always in combination with something else. And this is important because certain modifications have negative or positive crosstalk. And so, for example, arginine methylation on histone H3 actually inhibits the enzyme methyltransferase from methylating K4 on the same protein, whereas arginine methylation on histone H4 actually stimulates acetylation of some residues that are downstream. And so there's all these different kind of hierarchies and relationships that chromatin biologists have been uncovering over the years. And so this brings up a really great question, right? Because, if we take the common proteomics approach to look at histone modifications, and here's a sequence of histone H3, and if we digest it with an enzyme that will digest it into smaller peptides, we can detect a lot of these peptides and the modifications on them. But if you ask me, well, do you ever see this modification with this one? I can't tell you if I ever see them together because I lost the connectivity once I digested the protein. And since many of these modifications turn genes on or off, this is a really important question that needs to be studied in depth. Do you ever see antagonistic modifications together on the same histone tail? Or which combinations do you see together? And is this really a code? And if you think of a code, is there some kind of way to interpret this code? And so over the years, we've had many iterations of developing, just different chromatographies and mass spec-based approaches so that we can get to the endpoint of looking at, the first 50 or 60 amino acids on the histone tail and all the different combinations and quantifying, detecting, and quantifying all the different combinations of modifications that occur together. And so we, just kind of show you a little bit, not going to get into the technical details, but, we can take a histone such as histone H3, do a separation with, this kind of newer chromatography called HILIC that we've kind of developed over the years. And it separates out the histones by their PTMs. And that allows us to then select certain modified forms of the histone for fragmentation, which will allow us to get their sequences. And at the end of the day, for one kind of peak here, we can see that this is the major histone code that's present with the multiple combinations of modifications at the different lysine residues. And so, we've been able to kind of do these experiments, but as we're generating this data, what we will really want to understand is which histone modifications really don't care if they're found together or not and are kind of independent, which ones have kind of, interdependency or positive interplay, they like to be found together, or which ones really do not like to be found together. And so now over the years and looking at, many different cell types and conditions and things like that, we've now found, that there's about 2,500 histone H3 forms across 3 different variants. And there's certain modifications that really like to be found together and other ones that really do not like to be found together. And several rules and hierarchies. And you might think 2,500 is a lot of forms, and it is, but theoretically it should be millions. And so, there's definitely rules and hierarchies that have to be obeyed. And that now we're starting to, and I'll show you in the second half of the talk that some of these really do matter. These combinations of modifications together do matter. Another area that we're just trying to increase or enhance is really the throughput nature of these types of analyses. And typically the experiment is that, you take your histone or any proteome sample and you separate out the peptides by some kind of liquid chromatography, usually C18 reverse phase, into the mass spectrometer. And in our hands, this ends up being kind of the bottleneck because complex proteomes usually require long gradients, 2 to 3 hour runs to sample everything. If you have a bad sample, you just ruined your columns, so that's going to ruin the run. And let's say I have 200 samples I need to run. The chances are I'm probably not going to run them on the same column. I'm going to have to change out the columns, and this is going to create batch effects. So for us, we were always wondering, well, I wish we could get rid of this type of step and just spray directly in the mass spec and not have to deal with this headache, because also HPLCs are finicky as well. And so we decided to do that. And so we use this instrument called a nanomate, which is a direct injection instrument. It picks up a sample about 1 or 2 microliters and can spray it for even up to about 5 to 10 minutes at a time. But what we decided to do was not just spray in a sample, but also tell these hybrid mass spectrometers, such as an Orbitrap fusion, to do the separations in the gas phase, because we're kind of getting rid of the separation in the liquid phase. So we're going to lose some resolution. But these instruments have multiple mass analyzers and functional other optics that allow us to do some types of fractionation in the gas phase. And so, we decided to do a proof of principle experiment where we took BSA, we digest BSA with trypsin, and if we run it by like a 30-minute, 40-minute run with a nano-LC liquid chromatography and get about 70% sequence coverage of BSA, then we know that our sample, our instrument is ready to go. That's kind of our QC, and it's ready to go, lets system stability, and we're able to run. And so we took BSA, we digested it, and then sprayed it in the mass spectrometer for about 20 to 30 seconds, and just let the instrument do different separations of the ions in the gas phase. And what we found is we got 61% sequence coverage without even optimizing. So that was really kind of very encouraging. And so we decided to make kind of a gas phase fractionation scheme for all the histone peptides that we're really interested in. And so, we kind of know a lot of these peptides, or at least their families, and we can kind of estimate windows that would include their modifications. And we're going to allow the mass spectrometer to kind of do the separations in the gas phase of these different masses, rather than try to get liquid chromatography separation of all these different peptides. And there's, things that we have to do to just increase the signal, because once you lose liquid chromatography, you lose a little sensitivity. So there's things we can do to kind of accumulate single signal in these different parts of the instrument. But at the end of the day, we did a proof of principle experiment, this direct injection of a sample versus our nano liquid chromatography, 90-minute run. And we took a peptide library that has synthetic histone peptides of different types. There's probably about 75 peptides in this library. And we ran them both by the direct injection and the normal run. As you can see here, most things are pretty close in intensity. There's a few things that are outliers for sure. But what we actually found was that the direct injection actually gave us a little bit more accurate results. And that is because with liquid chromatography, there's some peptides, such as these K4 peptides, histone H3 K4 lysine methylation peptides, that don't stick very well into these columns. And so sometimes we see them, sometimes we don't. But with direct injection, there's no columns, so we see everything. We took a brain and liver sample from mouse, real sample, our normal nanoLC-MS 90-minute run versus, I didn't go over its acronym, but basically it's direct injection, mass spectrometry for 20 seconds, the same sample. And what we get was really, really similar results. And so now we're getting to the point where, we can run, 300-400 samples a day. I don't know if we've really hit that many, maybe just a few times, but we have the capability to really go into large scale experiments, go into clinical samples. And so I think right now we're at a really exciting time that we can quantify, depending on the cell type, somewhere around 150 or so, maybe a little bit more histone PTMs in less than a minute. Lastly, technology-wise, I just want to talk a little bit about some work that kind of is outside the epigenetic realm, but it's still, I think, very important, very interesting, and that's RNA biology and RNA modifications. So obviously, there's a lot of different RNA modifications that have been popping up over time, and now we're starting to see that these modifications have definitely biological function in various areas of biology and disease. And the one way to look at RNA modifications is typically with next generation sequencing approaches. However, with these modifications, oftentimes you either have to use antibodies to select one of the modifications and enrich for it before you sequence, or there's some kind of chemical kind of indirect method to detect the modification. And so we always thought, well, mass spectrometry would be great to be used to detect RNA modifications. And so we take an RNA sample and digest it with an RNase and ran in the mass spectrometer in negative mode. So there's a lot of technical issues here, but we kind of worked out things over a while. And what you're seeing here is an LC-MS run, and every little spot here is an RNA peptide of different sizes eluting out. And we're able to optimize the fragmentation of the RNA species. Typically, they're anywhere from 6 to maybe 30 oligonucleotides long. And if there's modifications, we can detect those in the mass spectrometer. We did have to create a new computational workflow for this because there's no shotgun RNA omics kind of software out there to be able to do large scale types of analyses of mass spec data on RNA, fragmentation data. So that was kind of something we really had to put a lot of work in, but we now have a nucleic acid search engine And this allows us to kind of map all these little spots and sequence them and say, oh yeah, it's this RNA with certain modifications. And so this is kind of screenshot of one of the data sets. So, got pretty good fragmentation of the RNA species. When we have modifications, they're very easy to pick up because they give us these mass shift changes. We're able to detect, somewhere around 30 different RNA modifications pretty easily. We still have a long way to go with this. Typical sequence coverage is like 40 to 60%, which is kind of low. And this is because we just need to find more nucleases or better combinations of nucleases or something like that. Because there's a lot of sequence space that is not quite covered really well, especially if you have several repeating units of the same oligonucleotide. And so, but it's a start and we're pretty happy with this, that we're able to do this. There are 2 programs out there that kind of can take one spectra at a time and you can map like RNA modifications with these one spectra at a time work engines. But ours is literally like a large scale kind of engine where you could dump a whole raw file with 100,000 spectra in it and get back your data. But in kind of comparing what we're doing to the previous examples, we're doing a pretty good job. So we get a lot more spectra identified and a lot longer length oligonucleotides identified as well. So this is kind of a first paper on this new area in the lab that we hope to keep kind of building on. But I think it's one that is going to be really important, and hopefully mass spectrometry will kind of be seen as a very complementary approach to other RNA sequencing approaches. So now for the second part of this talk, I just kind of want to speak a little bit about how we're kind of using that technology, especially the Histone PTM technology, to really try to understand a very specific type of cancer. And so for the last few years, my lab has been really interested in soft tissue sarcomas. And so sarcomas are pretty rare tumors of mesenchymal origin. And so you can see how rare they are just by comparison of the US breast cancer incidence or lung cancer incidence versus the soft tissue sarcomas. And so if you take a look at the y-axis, you can see that breast and lung cancer are just way more frequent cancers than sarcomas. But there are a lot of very different types that have been identified, and these are affecting a lot of patients. So these are important types of cancers to study. And just because something is rare, not that frequent, it doesn't mean that it's not important. And so obviously it's going to be important to the patients that suffer from these rare cancers, but it's also an opportunity to really learn more about some of these rare cancers and the common kind of mechanisms that can occur. And in many sarcomas, there are, and in many cancers, as I kind of mentioned before, there's a lot of mutations in chromatin-modifying enzymes and interacting proteins. And so 2 of the complexes that are highly mutated across many cancers are the SWI/SNF and the polycomb group of complex. And so you can see here the SWI/SNF, highly mutated. It's probably one of the more highly mutated complexes across many different cancers. Polycomb as well is highly mutated, especially in a lot of blood cancers. And these are kind of antagonistic in a way, functionally, proteins. And so polycomb contains the enzyme EZH2, which methylates histone H3. And so typically this is a silencing mark, and it keeps kind of a closed chromatin gene silence. So you can imagine if there's a loss of function mutation, this may lead to a more open chromatin. SWI/SNF is the opposite. It's an ATP-dependent remodeler. And so it slides nucleosomes around, allows access to underlying DNA needed for transcription. So it keeps an open chromatin, but you can imagine that SWI/SNF, loss of function mutation could lead to a more repressed chromatin. And so, these are probably the most well-represented of the chromatin enzymes that are being studied in various different cancers. But, as I mentioned, in mesenchymal tumors, there's a lot of different types of very interesting kind of chromatin abnormalities. And so we do see the SWI/SNF loss of function or even gain of function Polycomb loss of function, we see that as well. There's a whole host of sarcomas that have many different types of rearrangements where they create fusion proteins. And some of these will fuse like a histone acetyltransferase to a transcription factor, a demethylase to a transcription factor. And in many cases, it's not really clear what this how this fusion is driving, the cancers. There's even mutations to the histones themselves. And so in some bone cancers, you'll actually get, a histone where there's like a lysine mutation to a methionine. And so all of a sudden you wipe out methylation at that lysine residue, in these cancers. So there's all this really interesting kind of, chromatin biology that's being disrupted in these various cancers. One of the soft tissue sarcomas that we've been getting really interested in is malignant peripheral nerve sheath tumor, MPNST. And so as its name kind of implies, it's a sarcoma of nerve origin, peripheral nerve origin. It's both sporadic and syndromic. And so it can kind of progress from a related neurofibroma, or it can just kind of sporadically occur. It's a pretty rare tumor, probably looking at several hundred cases a year, something like that, but has very poor prognosis in children. And in fact, you can see there's less than 50% 5-year overall survival rate. Not a lot of options to treat patients, as many of them are poorly chemo responsive. And so, surgical surgery and kind of radiation are really the main staples to treat this type of cancer. There's a lot known about the factors that can drive it. And so about 50% of the patients have a mutation in one of the cyclin genes. And so that's kind of a one set of MPNST, but about the other 50% have a mutation to SUS12, which is a component of the polycomb-repressive 2 complex. And I mentioned this complex before, right? This is an enzyme complex. It methylates histone H3. This is a silencing mark. So you'll get this on and find this on the parts of the regions of the genome that are silent, that are not expressed. And in fact, because of this, it's this histone H3 K27 is what is used in the clinic to kind of separate out different types of MPNSTs. And so here's a MPNST from a patient tissue sample that has a polycomb retained, and so it has a different mutation. And so you can either see the staining for histone H3 K27 trimethyl, whereas here's an MPNST that has a polycomb loss. And so you do not have the H3 K27 trimethyl, and you can see that you lose the staining. And that's what kind of pathologists use. However, K27 trimethyl is probably not the best marker for this or other cancers. And that's because it occurs in a lot of other cancers. And so it's not specific to this one cancer. And in fact, there's some cancers, such as melanoma, that are actually at the pathology level similar in scope to MPNST. And so trimethylation is really not a great marker to differentiate, some of these kind of closely, not related, but closely pathology-wise, I guess, closely related types of cancers. And so as we kind of started off in this project, we kind of had a few different questions. One, we know PRC2 is the major loss, right? It's a loss of function mutation. We know K27 trimethyl is going to be decreased, but how does this PRC2 loss further affect the epigenome? How does that reprogram the rest of the epigenome? How does it respond to that? So we're going to use our histone proteomics type of technology to try to understand that. Additionally, anytime you're affecting the epigenome, you're going to affect gene expression, you're going to affect protein expression. So we're going to use quantitative proteomics, kind of a little bit standard approaches to understand which cellular pathways are affected by the PRC2 loss. And this was all driven by a resident pathologist, postdoc fellow in the group, John Wosick. And at Penn and many other institutions, once you have tissue sample that has been taken from a patient, oftentimes they're stored in these tissue banks. But the one issue is that they're stored as formalin-fixed paraffin embedded samples. So they're these little kind of samples embedded in wax, right, and cross-linked. And so there's a ton of these at Penn, my former institution, when I was there. And so John had to really kind of convince himself that he could get high quality samples out of these wax kind of stored samples, high quality proteome, high quality epigenome, histones. And so he really worked hard to kind of optimize, the proteomics from FFPE samples. And so he took a fresh piece of liver, and then he formalin fixed, paraffin embedded a piece of liver and extracted out the proteins. And just a really quick run showed that he can get about the same number of proteins, which is great. And the abundance of the proteins are about the same. There's maybe a little small decrease in abundance of some of the lower level proteins in the FFPE sample. And that's because you can't fully cross-link or reverse the cross-links completely in these samples. So it is a little bit low, slightly lower quality, but we were pretty happy with these results. And from fresh to the fixed, you don't really get a loss or a bias in anything, right? We don't see like membrane proteins or nuclear proteins being lost. You get a little bit of loss of everything. So, or very, very small though. So we were really happy with this. And John went back and then, did some tests of, well, how far back can he go in terms of stored samples? And I think he even went back about 12 years. And you could get very, very similar numbers of proteins now with a little bit longer gradient, deeper proteomics, get about 5,000 proteins or so pretty easily. And their abundances were all very similar. So we were really happy with this data from, real FFPE samples. And then with histone modifications, that we took that fresh and fixed liver sample and just looked at the top, 50 or so, 100 or so histone marks. Very, very good correlation between fresh and fixed tissue. And if we took cells from patients that have the mutation versus the FFPE tumors from patients and looked specifically at the K27 modifications, the methylations, very good correlation as well. So we thought we were in a pretty good position and should be able to get high quality data off of this. And so John did an experiment where he just took neurofibroma, the MPNST with polycomb loss, or the MPNST with the polycomb that's retained. As you can see, there's something very different about the MPNST with the polycomb loss. And here's a volcano plot of the polycomb loss versus the polycomb retained MPNST samples. As you can see here, we do see the loss of a lot of the K27 methylations, which we would expect. It's a polycomb loss. These are kind of shown here for K27 dimethyl and trimethyl. The neurofibroma, polycomb retained, polycomb loss. We can see for the dimethyl and for the trimethyl, huge decrease in those modifications. And a lot of them are found with K36 dimethylation. So this is a silencing mark, this is an active mark. So that was really something interesting that specifically we saw the loss of this dual mark together. There were other things that were lost too though. So I mean, you can see here, besides the K27, we lost K20, and then we also gained some things as well, histone acetylation, K36 dimethylation by itself. And so these are the things that were upregulated in the polycomb loss. So a lot of acetylation, as well as the K36 dimethylation. This is the active mark. These are all active marks. And in fact, a lot of these right here that are circled, they're actually histone H4 acetylation. So you have lots of different combinations of histone H4 acetylation that occurs. In the polycomb retained, here's like the normalized level. Polycomb loss, you get an increase of many of these specific combinations of acetylation on histone H4. And so a little bit deeper dive into the K36 methylation data, we can see, like I showed you down here before, we can see these peptides that have K27 mono di or tri with K36 dimethylation together. And the polycomb retained, levels of K36 di with either unmodified mono di or trimethylation levels are right here. And we see a good spread of these different K27 methylation with the K36. So you have the silencing and active mark together. But in the disease state, you lose the K27. You basically have K36 levels by itself with the K27. This is the unmodified. And so what this data was telling us was that in the normal state, you have parts of the genome that have K27 methylation. You have parts of the genome that have K27 and K36 and other active marks together. But in the disease state, you lose the K27 and you have just active marks kind of overriding. The really interesting thing as well is that we actually found that K27 dimethyl with K36 trimethyl are good markers, even better markers than the K27 trimethyl alone for MPNST. So here's a polycomb retained sample. We see both the K27 tri and di. Polycomb loss sample, we lose both of them. And not only do we lose both of them, this is really specific for MPNST. I mentioned that there's a lot of other different cancers that lose K27 trimethyl, but a lot of these other cancers, such as glioma and epidermomas, these don't lose K27 dimethyl. Only MPNSTs lose tri and di together. A lot of these other cancers may lose tri, but you'll see the good staining for di. So John actually started going back and trying to get pathology labs to use both of these markers now instead of the single one. And so we found out a lot about the epigenome with our quantitative proteomics approach. We actually saw that you go from a fully silenced state to a more active open chromatin state, as kind of shown by the histone active marks, acetylations and K36 dimethylations that increase in abundance. But what about the proteome? What's affected by this? And so doing quantitative proteomics, top 5,000 proteins or so. Here you see a volcano plot, log fold chains, polycomb loss or polycomb retained. So these are all the things that are gained in the polycomb loss. And you can see these are all things that are either related to cancer growth or things that are not fully unexpected, things that maintain an open active chromatin or active transcription. And so the histone mark data told us you go from silencing to open chromatin, and the proteome data tells us that, yeah, we see that as well. You have SWI/SNF and other proteins that maintain an open active chromatin and transcription that are upregulated, as well as things involved in cancer progression. However, you see that there's also a lot of proteins that are downregulated too. And so what are these? Well, these are all proteins involved in the immune response, interferon signaling, MHC protein complex presentations. And you can see this in the data real easily if you just look at the MHC proteins, class 1 and class 2. In the polycomb retained, you get signaling for all of these. There's different patients, so you get signaling for a lot of these, but the signaling in the polycomb loss patients is much lower. And you can see this bird's eye view by just immunohistochemistry of the HLA molecules. You can see in the polycomb retained, you get the signal, polycomb loss, you lose it. Same thing here, the signal is a little lower to start with, but you see the signal here and you lose it over there in these patients that have the polycomb loss. And so, although we go to open state and there are, chromatin proteins and growth factor signaling that is upregulated, there's this whole repertoire of immune signaling molecules that are downregulated. And this is a really interesting observation because, how does losing a repressor like PRC2 lead to more repression? How are we getting repression of these to start with? Why aren't we just getting runaway transcription? Everything going up that more PRC2 is found. And so John looked through the data really carefully and saw that there's a protein, DMNT3A, that was upregulated as well. And this is a DNA methyltransferase. And so if you look, and after we did bisulfite sequencing of samples, we could see that in the polycomb retained cells, you have a certain level of DNA methylation. The polycomb loss, it actually goes to a hypermethylation state, which is consistent with upregulation of the enzyme responsible for this. So you can see that here in the polycomb retained, you have a low level of, or a certain level of DNA methylation, and then blue indicates a higher level of DNA methylation in the polycomb loss cells. And if we look at, well, what are these genes that are found on the promoters and enhancers, this methylation, these are all interferon and the MHC presentation. So it's everything we saw before in the proteomics data. So this is kind of the mechanism for why we're getting downregulation of the immune system, is that DNA methylation is actually increasing. So it's compensating for the polycomb loss and is making these patients more immune compromised to allow for the cancer progression. And so what happens if we can manipulate some of these pathways? And so, at least in cells, we can restore polycomb, right? So it has a SUS12, it's mutated. What if we added a SUS12 that was active again? And what if we knock down some of these enzymes that increase the active histone marks here, such as NSD2, which is a K36 methyltransferase. K32 dimethyl is upregulated in the disease state, but if we knock it down, and what if we use DNA methylation inhibitors as well? And so I mentioned that, the mass spec data shows that the silencing mark K27 di or tri is found together with K36 di. So this is a normal state. And in the disease state, you lose the K27 di or tri. And so you just have the K36 by itself. This is the disease patient state. What happens if we can restore the methylation here, or if we can further remove the K36 methylation? That's what we'll try to do in these just cell models. And so the first thing we did was actually added back SUS12 to these patient-derived cell lines. And so you can see that we can blot, and from the control, we have SUS12 back. It's really interesting. If you IP for EZH2, one of the components of the polycomb complex, in the patient-derived cell lines that have the SUS12 mutation, you can never get back the full complex, and that's probably why it's compromised. But if you add back SUS12, you can then IP and get back the entire complex. And of course, if you get back the complex, it's functional. And so, you can see that K27 mono, di, and trimethylation levels all increase. You can knock down NSD2. So here's NSD2, and we can knock it down. It's the K36 methyltransferase. We can knock it down and get to this state. As you can see, the knockdown reduces the K36 dimethyl. Now, if we do quantitative proteomics of the whole proteome from these two states that we've this state and this state that we've manipulated. So this is the SUS12 add back or the NSD2 knockdown, and kind of plot the data that are differentially regulated by these two different types of enzymes, we can see that they're the same proteins, or they're very, very strong correlations. And many of them are the things we saw before. And so indeed, we see both K27 di and trimethyl on the same genes. With regards to cell growth, if we knock down NSD2 in polycomb-retained cells, it doesn't really matter. They don't care. They're not very sensitive. But if we knock down NSD2 in the background of the polycomb-lost cells, these cells have a cell growth defect. We could play some other games too. Instead of knocking down the enzyme, we could actually remove the mark by putting in this transgene, this histone H3 K36 methylation transgene, so it can't get methylated. In the polycomb-retained cells, so these patients have these cells have a different mutation. It doesn't matter, if we lose the K36 methylation levels, it doesn't really do anything to cell growth. But these cells are very sensitive to losing the K36 methylation levels by doing this, very similar to the knockdown of NSD2. Using a DNA methyltransferase inhibitor, I mean, this is a big hammer. There's a lot of DNA methyl transferases that will affect cell growth. So if we use increasing concentrations of this DNA methyltransferase inhibitor to MPNST cells with polycomb retained, eventually you can kill the cells. But you can see how much more sensitive the cells with the polycomb loss are to the DNA methylation inhibitor. And you can get re-expression of the interferon genes by adding this DNA methyltransferase inhibitor to the cell culture. So you can see all the interferon and immune system proteins coming back, and there's this also will end up causing re-expression of endogenous retroviral genes as well. And so with these manipulations, we're learning a lot about the system. You lose K27 methylation, you have open active chromatin. The genome maintains this by upregulating proteins that are needed for this and to drive transcription. However, at the same time, the patients are being immunocompromised because DNA methylation is taking over and silencing genes that are needed for antigen presentation and interferon response. Unfortunately, you can't do a lot of these things in a patient, right, as a treatment, right? How do you, restore function in a patient? And there's no NSD2 inhibitor presently available. There's DNA methylation inhibitors though, but you can't kind of do these as a treatment of patient. It was a good academic exercise, but not for patient treatment. But there are other histone marks that are changing too, and specifically acetylation, went through the roof in these cells. And DNA methylation and histone deacetylases do share a shared mechanism of action. So we thought maybe this could be something to explore. And the idea was that, in the polycomb loss cells, you have high levels of histone acetylation. What if we just drove these to cytotoxic levels by adding an HDAC inhibitor? And so if we take polycomb retained cells and look at H3 and H4 acetylation and add an HDAC inhibitor, panabinostat, say you get an increase in the acetylations of both. Polycomb loss cells already have like a higher level of acetylation, even compared to the polycomb retained cells with the HDAC inhibitor, which is pretty interesting. And then when we add the HDAC inhibitor, those levels go through the roof. So we're definitely getting to this area. So what happens to cell growth? This is the last slide I'll show you of data, and then I'll wrap it up. So if we have polycomb retained cells and we add increasing concentrations of this HDAC inhibitor, panabinostat, here's kind of the kill curve for that. These three cells in red have the polycomb mutation. And so you can see these are all much more sensitive to the increasing concentrations of panabinostat. And in fact, you can get the re-expression of a lot of the immune response proteins, HLA proteins by addition of this panabinostat. So all these mechanisms are converging. If we took one of these cell lines that had the polycomb mutation and we added SUS12 back to it, remember we did that before in those experiments, now these cells become more resistant to the increasing concentrations of the panabinostat. And if we do the combination kind of therapy, add increasing concentrations of panabinostat and a DNA methyl transferase inhibitor, you can see that the combinatorial kind of treatment, kills the cells, a lot easier than the cells that have the mutation versus the polycomb retained cells. And so, taking kind of a quantitative proteomics approach to look at the histone marks epigenome as well as the proteome really allows us to understand a lot about this system, and maybe potentially points to a few new areas that could be further explored for therapeutic routes. And so, kind of in summary, PRC2 loss, the K27 methylation is by far the largest loss epigenetically, but the epigenome kind of is reprogrammed. And so we see increasing histone modifications such as acetylation and K36 methylation kind of reestablish themselves as well. And along with that, another silencing mark kind of compensates for the loss of the K27 trimethyl, the DNA methylation. And so these might all be potential routes to look at some point for new potential therapeutics. But there's still a lot to understand. And so, this is ongoing work that we hope to continue to really try to understand relationship between DNA methylation and histone modifications. We have some ideas of what might be going on here. And we think we can get at that with some ChIP-seq experiments and other things. And then really this whole, new area of the immune response. Why are we getting the decrease in immune response epigenetically is something that really needs a little bit more work, maybe in a mouse model. We now have a collaborator that has a mouse model for MPNST, because it may be the combination of epigenetic inhibitors with the immune response kind of inhibitors, checkpoint inhibitors or something like that could be the route to go in the future. So with that, just want to thank all the people that are involved in different aspects of technologies. Always had a lot of great students and postdocs over time, and staff as well. And just, single out John Wosick, who did a lot or most of the work with the MPNST cancer as well, and funding from different sources. Thanks again for having me here, and I'd be happy to try to answer any questions if there are any. Thank you.
Great, thanks, Ben. That was an awesome talk. So if you have any questions, please unmute or go to the podium and ask Ben your questions. Maybe while others are thinking of that question, maybe I'll get the ball rolling. One question I had was, have you profiled epigenetic drugs? You showed panabinostat, but have you profiled there's so many compounds that have epigenetic effects? You could then see if any of those reverse this effect that you see in sarcoma, or you could profile them just to get the best match, sort of.
Yeah, well, we haven't done that specifically in this with this sarcoma, with this MPNST. We kind of just went for, there are a handful of HDAC inhibitors that are clinically approved. So we, we kind of went after one of those, the panabinostat. However, I've always kind of wanted to go back and profile. There's so many different HDAC inhibitors, and they must all have slightly different, histone or non-histone targets, or, severity of inhibition of HDACs. And so I've always kind of wanted to go back and kind of do a little bit larger scale type of experiment, where we can take something like, I think the company Celic offers a histone inhibitor library, maybe has 200 different compounds on that, or maybe try to collaborate with, the Toronto SGC, consortium, because they have a lot of other HDAC or a lot of other epigenetic inhibitors, and really just profile everything to kind of get a really, quantitative atlas of, the different responses, not just epigenetically, but maybe throw in other things as well as cell growth and different types of things like that. I think it's a great idea. It's a pretty big undertaking, so we'll have to kind of figure out how to do that well.
Yep, awesome. That'll be super useful though for us.
Yeah.
Hi, this is Tom Crapola. Nice talk. Both parts were interesting. I'd like to ask about the second part regarding the role of PRC2 in keeping interferon responsive genes on, which is a bit unexpected. Do you have parallel patient lines from normal tissue to say whether the PRC2 change is in fact the operative difference in terms of distinguishing between something else perhaps affecting PRC2 and the interferon response in parallel, as opposed to the PRC2 actually keeping interfering response genes on?
Yeah, no, we don't. That's definitely something that if we can get back to it, it's a very good question. And it's definitely something we've kind of wondered too, wanting to get back to that whole immune response kind of angle. That's something that we would take very close look at. And if we can get the right samples, I think we could get a good answer for that. But we haven't not done those experiments yet.
It's really interesting. I guess as a follow-up, you mentioned the question of immune function in sarcomas. Is there other data to say that actual interferon response gene expression in the tumor has prognostic importance in terms of the sarcoma? I mean, is this something that actually is a pathological mechanism?
Yeah, I don't think it's been well studied. I mean, from the few people we've talked to in this area, it's definitely not, something that's on people's radar. Yeah. So yeah, no, it remains to be seen.
Thank you.
I think Carolina had a question.
Yes. Hello. Hi, Dr. Garcia. Thank you for your presentation. I have a question. So you mentioned, the rate limiting step was the LC, which indeed is the case. But I have seen other approaches where they replace it with ion mobility. Are you planning to maybe use some ion mobility in the future? I know at some point there was not a lot of software, and there were some duty cycle limitations, but there are many labs now that are using the team stuff for proteomic gradients. So I was wondering.
Yeah, so it's a really good question. We've already tried some ion mobility like FAMES, like in the front end. The hope, I think, for us was really that we were wanting to, without the liquid chromatography, the worst thing for us is that there's several peptides that are like isomeric modified peptides that have multiple lysines. So you'll get one peptide that has the modification on one lysine, the other peptide has modification on the second lysine, but they kind of co-elute. If your chromatography is really good, you can kind of separate them. So we kind of work at that. But without liquid chromatography, now you're just spraying them all together. They have the same mass. And so we're really hoping that, the FAMES, we have a FAMES kind of source, could be, something that would help there. However, we didn't see kind of the separation that we were wanting out of the FAMES. We have not tried something like timsTOF or like a real ion mobility source. That's kind of a little bit more outside of what we can do. But I think you're absolutely right. And one of the follow-ups from this initial work that we did in Histones is that we want to kind of do things at a more complex level, whether that's proteome, I'm not sure, but maybe like a fractionated sample or maybe like an IP or something like that. And I think there, some type of ion mobility would be essential. I think what we found for the histone modifications, we can kind of get around it. It wasn't essential. We just kind of, can use prior knowledge and some math, and we can kind of estimate the modified forms if we have to. But for a really complex sample, you're absolutely right. We're going to need an ion mobility. So, hopefully at some point, someone can kind of couple those together and do it.
Cool. Thank you.
Any other questions? not, I would thank Ben again. Oh, Brian, do you have a question? I saw you come in.
Oh, no, I just thought, I wanted to thank Ben as well. I thought it was a fantastic talk. As an old Chromatin guy, it's just so much fun to see how this has gone along. I mean, thanks so much for coming to our seminar.
Yeah, no, thanks, Brian. Thanks.
Really
Shriram.
enjoyable talk, and the power of the mass spec, you know, it's really amazing how that's creeping up everywhere. So this should be it was a very big interest to us, and I hope you're able to visit, you know, when time permits.
Yeah, definitely. You know, I've actually never been to Michigan, University of Michigan, so it'd be great to come visit one day.
Well,
Sure.
consider this to be an invitation, and thank you very, very much. And Shriram, I didn't want to steal your fire. Go for it.
No worries. Yeah, that's what I would have told Ben too. You know, like once things gets better, you should definitely come visit us.
Yeah, sounds great. But thanks for having me, everybody.
Yeah, thanks, man. Yeah.
All right,
Really great. Thank you.
thanks. Bye.